Ok, parfait.
I always wanted to be Beethoven when I grew up.
Welcome to the Night Science Podcast,
where we explore the untold story of the scientific creative process.
We are your hosts.
I'm Itai
and I am Martin Lurcher. Today with us is Ellen Rothenberg, and we're very excited to talk to you, Ellen, about your night science. Ellen, you received many honors. And just to give a few examples, you're a fellow of the American Academy of Arts and Sciences. You were recently elected to the American Association for the Advancement of Science. And what I personally find extremely impressive is you also won the 2016 Richard P. Feynman Prize for Excellence in Teaching.
Absolutely. And I think what is actually even more impressive is Ellen's research, which has made her, I think, one of the leading scientists of our time. In her research, she studies the mechanisms of vertebrate development, and more specifically, how it is that different classes of T cells develop. On a personal note, I can say that I've met Ellen at many conferences, and the thing that sticks in my mind the most is just her infectious energy and enthusiasm for science. And so, Ellen, it's really such an honor and a pleasure to be talking to you today on this podcast.
Oh, I'm thrilled to be here, and thank you so much for inviting me.
Ellen, just to get started, do you think there's any aspects of your training as a scientist that influenced your development in terms of becoming a creative scientist?
Well, my training as a scientist sort of ironically began before I was in college because my parents are both academics and they always encouraged my interest in science from a very young age. But at the same time, they infused it with an interest in the arts and literature and history so that I was brought up sort of immersed in an artistic culture. And I always wanted to be Beethoven when I grew up. So I think that the sense in which the emphasis was placed on creativity really came from long before I started to get formal training as a scientist. I don't know how closely I've succeeded in bringing creativity to science, but I hope I've done some.
In what fields were your parents in academia?
They're in social science. They were Franklin D. Roosevelt type economists, but they were very interested. My mother was a superb classical pianist. My father also was interested in classical music and art. And so we had a very strong discourse in the family about art and creativity and what it took to be creative. So going into science, that still was very much part of my set of goals, I think. And I don't think that anything that I learned in Harvard or MIT was able to completely wipe that out.
No, that's fantastic.
It was a joke. I had marvelous teachers at Harvard and MIT. So that was just a joke.
Can you distinguish that mode of creativity that you learned as a child or as a teenager in your work now?
I think there's just a sense of great dissatisfaction with doing something that other people are doing. It makes me feel very claustrophobic if we're basically following someone else's program. I'm not sure whether it's doing a great service to my students and trainees, but it certainly does keep shoving us against the boundaries.
No, and I think it's a great recipe to do groundbreaking research. There's this saying that what a lot of scientists do is they're enlarging a hole that someone else started digging. But if you want to find something really different, you have to start digging a hole somewhere else.
Yeah, or you can find the fracture planes where there might be some good places to start a new hole.
Yes.
I think it's a little bit more than that. There is a requirement to be very ambitious in one's thinking in order to see where the current structure of knowledge doesn't fit. And I think in this sense, not in all senses, but in this sense, I kind of agree with T.S. Kuhn and the structure of scientific revolutions, that a lot of times you do reconfigure the way you understand something when you realize that the prevailing structure of explanation doesn't fit. And the interesting thing about that is that what really helps is to be very explicit, very clear, and very detailed about what the prevailing structure does fit. And that way, when something really doesn't fit, you can see that fact. I think one of the things that I worry about with some younger people who've come into our field at a time when it was so data rich, they actually have a kind of a passive defeatism about that. They don't necessarily feel that they ought to be able to understand everything. And therefore, when something comes in that actually could be the key to a major reconfiguration, they don't see it because they don't have a well enough articulated sense of what it is that it would violate. Am I making sense?
Yeah. And I'm just wondering how we could kind of think about how it should be taught. How would
Well,
you convey to someone?
yeah, so for myself, one of the great educational experiences of my life was my decades-long friendship with a very strong-willed character, Eric Davidson.
I knew you were going to say
Yeah, well,
okay.
because he was very interested in gene regulation and how it specifies development, but he was extremely strong-willed, but also very, very ambitious, and he had an extraordinary grasp of a lot of knowledge about embryos. And we
Hmm.
used to have great arguments because he had a very large conceptual structure that he had crystallized over the years based on information from the way embryos do things in terms of developmental specification.
Right.
And so by having someone with such a clear, detailed sense of what things should look like, you actually really see when you've got a result that completely does not fit that. And it was fabulous to see the detail, the number of cases in which embryos are doing something one way and the hematopoietic cells that I was studying did something different, something that was not expected. So I think that that was a tremendous help. And I think these arguments were very valuable, these interfield arguments, because different biological systems actually optimize different things. And seeing that so clearly was a tremendous, tremendous help.
Yeah. And I think in general, the way the human mind works is we can only make contrasts. Something is only
Yes.
defined by how it's different from something else. I can imagine you thinking about development in the immune system and you had certain ideas, but only when you can see that they really contrast very sharply with
Yes.
what Eric Davidson is saying. All of a sudden you say, oh, actually, that is actually very novel and interesting because it's not what you would expect.
Yeah. And, it's that difference in the context of overall similarity. And it's also the detail in the picture so that you can detect not just one hole that somebody started digging, as you were saying earlier, but you can detect a whole structure of little cracks that give you opportunities to go through. Dose dependence, flexible timing, stuff like this. This is the heart and soul of hematopoietic development, but it is anathema to an embryo. So it's been very, very interesting there.
Yeah, but it's very interesting that you said that you need some strong-minded. I mean, you didn't call it an opponent, right? You called him a friend, right? But somebody who has a strong idea of, like you say, how things should be, so that you can contrast your findings against those ideas.
Yeah, it's a knowledge structure. I mean, I'm not saying that he's wrong about what he worked on, but it was such a detailed knowledge structure with so many pieces to it that fit together in that system that you could then see all of the places where your own discovery of something different would have implications. And so that gave you a lot of chances to go off and explore.
So far, I think you talked mostly about the background against which you develop your work. But when you actually come up with a new idea, with anything creative, do you think there's a specific method that you use to do that?
I wouldn't put it that way, Martin. I think most of it is inhabiting the data. So again, if you are really trying to get a full understanding of a mechanism, then every piece of data that you get either fits or it doesn't fit. And that can be, in the old days, we used to do experiments that were really hypothesis-based, and where every assessment of the samples that we made was based on a very focused hypothesis. Now we do a very large fraction of our work by sort of genomics methods, which give you this huge harvest of data, which wasn't explicitly part of your initial hypothesis. And it's there freely for you to play with. I think I drive the people in my lab crazy looking at genome browsers and looking at gene lists, because I'm always trying to cluster them and put them together into pathways that I think I already know about. And I start jumping up and down all the time when I can find something that looks as though it's showing us either a new conjunction or a failure of a conjunction that we thought was there. And it's always trying to live inside the data and use it to test the structure. One of the more exciting things that we discovered maybe almost a decade ago now was a mechanism for the autoregulation of this transcription factor PU.1, which we knew had positive autoregulatory binding sites in its regulatory systems. And so we expected that when the cells ramped up its expression, it would be going through this transcriptional mechanism. And then I got a systems biologist into my lab who did live imaging, and we live imaged the levels of PU1 transcription coming off a fluorescent reporter. And we found that the slope of fluorescence increase over time, which should have been the direct output of this transcriptional mechanism, actually didn't change when we thought this factor was positively autoregulating. So instead of that, what was happening was that the cell cycles were getting longer. And there was such a strong expectation that this positively autoregulated and that it should be doing it by increasing its own slope, but it didn't. And instead, it was working on the cell cycle length. And so how could that possibly make a difference? Well, the only way it could make a difference is if the protein was stable enough so that the cell cycle dividing the amount by half was actually significant for its total level. And sure enough, it turned out that this transcription factor in this context is rock stable, it lasts for days. And so in fact, it shows you that you can have an entire mechanism of differential regulation of the activities of different factors that collaborate together at different balances by playing with cell cycle versus protein half-life. And I mean, nobody had this idea before that I was aware of. So here we had this strong expectation. It was totally violated by what we saw. And we had to come up with another explanation, which actually turns out to be true. And so that was an example of something that was quite exciting.
So do you still remember how you had that insight, or someone in the lab had suddenly that insight that it had to do with this interplay with the cell cycle?
Well, no, I did. I mean, I looked at it and I said, where is this upregulation and what is happening to the cells that they're going so slowly? And then we realized at that point that we had to measure the half-life of the protein, because that's the only way that you could account for the buildup of the protein without increasing this. Somehow it had to be reducing its degradation rate. And when you see the cell cycle, every time you're watching a fluorescent reporter, the level gets cut in half by the cells dividing the level per cell. And so you could see the baseline creeping up, but you could see that it was creeping up basically because of this elongated cell cycle time. That was a place where the data talked to us and we were willing to listen. And I think the willingness to listen and to be excited when your expectation is violated is really crucial.
And I think that's beautifully put. You have a hypothesis, and if it's wrong, well, first of all, it takes a big person to admit that the evidence doesn't support the hypothesis. But let's say you even make it that far. There is this expectation that, okay, you would now come up with a new hypothesis. But I think what you highlight is such an important point, that there is this conversation with the data, that the new hypothesis is not just a random one that you just brainstorm. Like, you actually look at the data, and I like how you say that you inhabit the data. It's almost as though you're putting yourself in there with the cells and trying to come up with what's going on.
Very, very much. I know that everybody knows the story about Albert Einstein, imagining himself being a photon. But in a sense, when we look at data sets, that's what we should be doing, even if we're looking at molecular biology data. If you are this transcription factor in this cell's nucleus, how the hell do you figure out which of the million possible genomic sites you're going to go to in this particular cellular context? And we don't really actually have a good answer for that. We have all kinds of algorithms for looking at preferred motifs, but they work very poorly to predict where transcription factors in mammalian cells actually are found binding. And so, this really helps you identify what I think are really major outstanding problems in one's field.
I think it's really interesting how you say, when you say inhabit the data, I kind of imagine you focusing on a particular gene and imagining the world from that particular gene's point of view. But then you say that in contrast to the old days where everything had to be hypothesis focused, now you have these genomic data sets. So I'm wondering how it is that you can sort of focus in on individual genes given the wealth of data sets. It can tell you everything about all the genes. How do you do it?
Yeah, I think that that's actually a big challenge. And I think it's something that our field really hasn't come completely to grips with yet. I have a bit of an advantage because I've been a scientist for a long time, and I've been working on T-cell development for a long time. And so I started out at a time when very few of these genes were known. And so they have very strong personalities to me. They all have backstories, and I know I have a slightly easier time fitting new pieces into that structure. I think for younger people, it's really tough coming in right now. And there are these very well-intentioned tools like gene ontologies that are supposed to help. They actually make your blood run cold, how poorly they actually do to represent what these genes are actually involved in known systems. So I do think this is a terrible challenge. I really don't know how to cure this problem for people who are coming in where they're just hit with, 25,000 lines of Excel spreadsheet and data to 4 decimal places in them. It's tough. I think it's good to think of distinguishing features that make a cell and to basically find these landmarks in one's data sets and then classify into modules. I think the modularization is really important. What are identity features of the cell? What are functional program submodules? What are, for T cells, what are immunoreceptor selection modules in terms of gene regulation? And then you start to recognize these clusters, and you can track the clusters, but they're functionally defined for the history of your cell type, so that actually helps you keep track of them, regardless of what the gene ontology tool says that those genes are involved in some other cell type.
So that sounds like one of the tools that you're using is to just have a set of questions that you always ask.
Yeah, if they're not the question, they're there as the kind of the orientation questions, like, am I really in the room that I think I'm in? That kind of thing. And because sometimes you find that you're not. And sometimes you find some very surprising things about, for example, the way enhancers work, or the way T-cell precursors differ from innate lymphoid cell precursors with respect to their deployment of the exact same transcription factor, totally different places of the genome. Guess what? Expression of this factor is not sufficient to predict what it's doing, And so those orientation questions, is this thing, the sort of checkoff list, is this really doing what I expect it to be doing? That's really important right up at the start. And then once that framework is established, you can ask the question that you thought you were intending to ask in the first place.
Yeah. You said something else a bit earlier on that I also found very interesting. You said, I imagine that I'm the gene. How would I go about doing what I want to do? Do you have a different way of talking when you're in that mode?
I think I use way too many metaphors all the time. So
maybe that's the secret of your success.
Any number of different ways.
Yeah, and you couldn't write a scientific paper like that, right? You couldn't write an introduction. If you imagine being that transcription factor, it doesn't work in what we would call day science, right? But in night science, it's a fantastic way to kind of try to understand what's going on in that process you're studying.
Yeah, exactly. And I think that this is really true. I mean, we all suppress our individual voices as much as we possibly can in our scientific writing, which is too bad, actually. That was something that has changed. People used to actually be able to write as writers in earlier days, and nowadays we have to sound extremely similar to each other. But I think obviously one wants to be precise about what one is talking about. But these analogies, I think, help us understand where the prevailing structure falls short.
Yeah, Itai and I actually recently reviewed a great book about how science works, The Knowledge Machine by Michael Strevens. We really liked it. We think it really explains a good
Amazing.
deal about how science works. And what he's saying is that there's this iron rule of science, and that is, if you want to decide who's right, the only thing you can argue with is data.
Yes.
Right? So all you can bring into the discussion is data. And that's how we write papers, and that's how we give scientific talks. But then to actually get to that point where we have a story to tell, then you can do whatever you want. It's just in the papers that you have to take a step back and only argue with the data.
Right. And the thing is, the data give you the path to understanding something you didn't understand before, but they don't necessarily tell you what it is. There are a lot of different ways that it could be. And I think this question of climbing into the data sort of imaginatively is sort of to harness the structures our brains are built with for very intuitive ways of learning about the world, ever since we're little tiny babies, and trying to see if any of those intuitive modes of navigating are showing you anything about the data structure. Then, of course, the second you've articulated what the question is that you think is interesting and new, you obviously have to go and very rigorously design the experiments that are going to test that. And you have to be willing to pitch that idea out just the way you would pitch out any other idea. I mean, you have a very high bar for actually accepting a new idea.
Right. Mm-hmm.
But if you haven't thought about what the new idea might explain that isn't already explained, you will never be impelled to look for the new idea or to test it. So there is this dialogue back and forth all the time between the day and the night science, if you want to call it that. And that is important. The other thing is that being a scientist means constantly questioning everything, including your own pet structures. And I think one of the real disservices that our publicity does to the world is to talk about what we've learned as though we're asserting it rather than talking about it as something which is actually the best we could do with numerous attempts to do better. And that we never stopped trying to do better in terms of explanations. We never stopped trying to do better in terms of challenging our own presuppositions. And the essence of being a scientist is this, cherishing and curating the structure in order better to see where it might not fit, where it might have to be fixed or broken and rebuilt. So it's something that many people in their normal lives are not used to doing. And I don't think we do a very good job of explaining it to the public.
We don't explain it. And it's probably in part because, as you say, when we present it to the world, we have to do that in a robust fashion. And then the exploratory idea generation gets naturally hidden.
Yeah. Although I think in a way, COVID has been useful for this because people had to watch in real time all over the world as scientists grapple with something that they really did not understand at first, except for some basic principles, and sort of watch the process of trying to learn. And some people obviously have missed this entire dialogue, but others have been watching. And I think that others out there may have been learning much more about science than they knew before, about the process, about the way you suggest ideas, the way you actually test it against data, and the way that makes a difference to the world.
Yeah, but initially it was almost funny/scary to see, how people didn't understand this process of science. I mean, just like you said, like there were people saying, but like 2 months ago, the scientists were saying one thing and now they're saying another thing. So how can we trust these people? Right?
Yeah, well, yeah, exactly.
That's science, right?
Some people learned that science actually takes sometimes as long as 2 whole months.
So, Ellen, keeping with this theme, I wanted to ask you, how do you distinguish any idea that you have as sort of a better than the fray idea, a great idea that you want to propose to test? And since you mentioned Eric Davidson, I wanted to mention this story that, so I just starting my lab, and I had this interaction with Eric that actually I came out very sort of traumatized by it. But in retrospect, I guess it's what I needed to hear. I explained to him an idea that I had, and he didn't like it very much. What he said was, he said, "You see this peach? You know, he takes a peach. He said, what you're talking about, you know, your sort of idea, it's just the fuzz on this peach. And what I'm interested in is the peach. I want to know the peach, not the fuzz. You understand me?" And I was very traumatized, but he set me straight. You have to focus on a better idea. So how do you do it? How do you know when you have an idea, it's okay, it might be an interesting idea, but it's not really what you want to devote the next 2 years of your life to?
Well, I think I try to generate lots of ideas, some of which are smaller than peach fuzz, some of which are tough, nasty skin, and some of which may lead to the sweet meat. I think that you don't know how far an idea is going to take you until you follow it a little ways. And you can't get too emotionally invested in it when it's still on the outside. Now, it may be that your goal is to define the topology of the surface of the peach, in which case looking at the surface might be very important. I think, again, the issue in your conversation with Eric would have been, what is the question? But honestly, you have to follow it a little ways to see how robust it is before you see how far it takes you into understanding a large new problem that you hadn't been able to get at before. And one has to take kind of tentative steps bit by bit at the time. Now, we as scientists nowadays have a big problem, which is that we have to constantly oversell our work and our ideas. And we have to constantly describe it, say, to reviewers and funding agencies as though every experiment that we do is going to solve the greatest problem in the world and that it's absolutely guaranteed to come out this way. And we and the reviewers know this isn't true. I don't actually understand how we allowed it to get to this point. I mean, it certainly isn't true in the practice of being a scientist, but I think the fact is that you can come up with a lot of different thoughts and a lot of different avenues of investigation, which will be transformative sometime down the line, but you have to be allowed to develop them to get them to that point. I deplore the fact that we are being pushed into overclaiming constantly when work is at an early stage. That is the stage when you actually have the chance of starting something that will be transformative later on, but you have to let it grow and get stronger first and get it debugged first, to use a different metaphor. So this is a problem. And sometimes you have an idea which turns out to be dead wrong from the start. That's very easy. Sometimes you get an idea which is good for a small local problem, but it really doesn't have any throw weight when you go to other problems. And then sometimes you come up with something which you then recognize as being a mechanism that's involved in a lot of phenomena that you hadn't had an explanation for before. And that's really exciting. But you have to get to the point of testing that first.
Yeah. And you said at one point, I don't know how we let it get to this point. Do you mean that
we as the scientific community, I think we've collapsed. We're doing very poorly at this point, I think.
I mean, I see it too, that for some reason, sometimes sort of being playful, which many of us recognize as kind of like the first steps of projects, is being seen as a liability, is being seen as something that suggests bad science.
Yeah. And we write papers backwards, right? I mean, we write them as though we didn't know what we know at the end, which was actually the last thing that we learned.
And this writing of papers backwards, I think it many times it can confuse the next generation of scientists.
It does.
Right?
It does. And the courses that I've taught, often when we dealt with scientific literature, often we had to go back to papers from more than 10 years ago in order to come up with anyone who even articulated any fraction of the actual logic process involved in the research. So the writing of papers has actually started to obscure the scientific process. I think this is a problem, and I think that also as these younger people are now grant reviewers and paper reviewers, but the template isn't necessarily accurate. So I think that this is sort of screwing up our
Yeah.
ability to communicate properly what we do. But nothing that I'm saying should go against the idea of needing to establish things with extremely rigorous and extremely searching, critical
Of course.
experiments and tightly controlled measurements. It's just that you have to first have an idea of what's worth measuring.
Yeah. It's almost like we should all publish a supplement to any paper that we publish where we describe the process that led to the hypothesis that we then state in the introduction of the paper.
I think that's a wonderful idea. I wish we were allowed to do it. It's such a shame because publishing became so distorted by the need to fit what used to be 5 papers worth of data into one paper and then to have draconian page limits or character counts, so that you were actually forced to cut all the logic out of your papers in order simply to fit it into the character count. I think that that's like the last hurrah of the print journal phase. And now that we have online publication more and more predominantly, maybe, that chance, says a cynical Ellen, but maybe some of this could be relaxed a little bit.
I never thought about that. What if the rise of these tabloids, Nature and Science, with their, as you say, draconian page limits, has led more and more to the hiding of the underlying logic and more of just clear presentation of the results. That's it.
Well, but I'm actually afraid that the habits of social media would go further into this bad direction, right? Tweets, how many letters are you allowed, right? And so you have to summarize a paper in that amount of space. That's even a lot worse than 3 or 4 pages of Nature.
Yeah, you're absolutely right. I think that this is a serious problem. And one thing is I feel tremendously grateful that I've been a scientist during the decades that I've been a scientist, but I get very worried about whether I'm able to leave the world to my trainees as such an exciting place to do science as it's been during the decades that I've been one. It's been such an incredibly wonderful profession to be in these years, just absolute joy. And I hope that our trainees are going to be able to have a large piece of that joy going forward 20, 30 years from now. But I don't actually know what form it's going to take.
Yeah, but I think you're absolutely right. That's one of our most important tasks, to prepare the next generation of scientists for a creative and exciting career. So do you have anything specific that you do to mentor your students, especially for the creative process in science?
I don't know. I do my best. I've never had a large lab. The people who've come to work in my lab, many of them have been just absolutely wonderful. Each one is totally different from the others. And so there isn't a one size fits all. I think that I try to get to know their marvelous qualities, and I try my best, which is obviously imperfect, to create an environment in which they can build on their strengths in a kind of a dialogue with me. But every person is different, and they prefer different kinds of ideas, and they prefer to follow different kinds of research. And so thing number one is obviously to allow them to develop the highest success with their own preferred talents, and to follow their own tastes and not try to mold them in some way that is not necessarily what they want. But they are facing a very different world than what I came up in. And so I do worry about, where they're going to find the breathing space for the kind of creative thinking and exploratory science that I was lucky enough to enjoy.
And if I could follow up on that, what does a typical meeting by a grad student look like with you?
Sometimes it's more than 3 hours of crawling through the data and just brainstorming, throwing ideas back and forth. I mean, it's all different kinds of things. I don't have a disciplined way of having, sort of regular 30-minute meetings with each of my people. Okay, time's over. Time for the next meeting. It's not the way we do things.
And do you have a particular way to handle when they're frustrated? Perhaps the project's not going their way. I feel like psychology is sometimes.
Yeah, and we're all amateurs, right? Look, the thing is that I'm actually really optimistic about science. And I think that when an experiment just isn't working, I just tell them, look, this is what science is like. An experiment that you
Mm.
haven't had work before, it can take a while to get it to work. There are a lot of variables. Don't worry about it. Don't panic. You're not bad. It's just a process. You're going to debug it. Just be systematic about it. It'll work. When it comes to actually data that doesn't fit with what they expected, I try to let them see this could be really, really, really interesting. And so we try to actually get into it at that point. What else did you see in these experiments that came out that way? What else could that
Mm.
be meaning? What could that be telling us about the system? Sometimes it's exactly these times that we see these observations. They start out as being depressing. Oh, I didn't get the result I expected to get. Well, let's look at it. Oh my
Mhm.
God, this is even more interesting. This is much more interesting.
Mm.
Let's take a look.
Yeah.
And this is sometimes how we make discoveries. So I
Yeah.
think that if you actually reproducibly get a result, which isn't what you expected, sometimes it is a whole heck of a lot better than what you expected in terms of breaking new ground scientifically.
Yeah, like a discovery by definition has to be unexpected.
One hopes, one hopes.
Yeah,
Yeah.
yeah. But it's really interesting to me that you said you don't have a systematic way of how you interact with the students. But what I understood was that you actually climb into the data together with your students, right?
I
And
try
it's like
to,
you go
yeah.
on a discovery tour together.
I try to. I hope they feel that I'm doing that with them. Yeah, I mean, that's very much what I like to do. I mean, in my normal life, when we don't have COVID, I have a busy schedule and I travel probably too much. And so the students and trainees don't get to see as much of me as perhaps they ought
Yeah.
to.
So is there a typical way of how a project in your lab unfolds? I mean, do you usually start with a hypothesis and then just realize, oh, it's not working that way, it's going to be something else? Or do you just start by producing some data that you think is going to contain interesting phenomena and then just look at it?
Well, I guess a lot of the projects start in different ways, but I'd say that a lot of times, based on things that we've been working on for a while, there will be actually a hypothesis set up as a question where either answer is interesting. So is this working such and such a way, or is it working such and such a way? So you try to come up with experiments that would distinguish between those two answers. And so that way it's a lot less likely that you'll just feel that you failed. You actually
Mhm.
find out something. Those projects generate usually a lot of results, which are sometimes quite surprising. For example, the use of an enhancer for gene regulation, is it being used only to kickstart the gene expression at a particular stage, or does it have a continuing additive effect on gene expression throughout all subsequent levels? And so in order to pose the question that way, you realize what sort of problems, what sorts of technologies you're going to need to deploy in order to measure things with the accuracy. But then in the process, you find out all kinds of things that you didn't know. You look at a transcription factor perturbation genomics data set, and you see clusters of genes being affected that you didn't expect to see. And so this becomes a hypothesis generating mechanism in itself. So I think that things often end up in different directions than the way they started, but they usually did start with a question, is it more like this or is it more like that?
Yeah.
Mm.
You said there's different ways of how projects unfold, but to me, it sounds like whatever it is, you always approach it with a very open mind, right? So either you just have a question or you have multiple hypotheses. And so I think that's a great way of making discoveries, right? By just being open to what the data is trying to tell you.
Yeah, I definitely really dislike the kind of science where you have to do an experiment needing to get a certain result in order to be a success. I think I actually
Mm.
shy away from that. And I think that's caused me to shy away from clinically associated research altogether because the
Mm.
benchmark is so stark. You're
Mm.
not saying, is it this way or that way? You're saying, does this work better?
Yeah.
And
Mm.
there's only one possible answer, or else you're a failure. And to me, that is not an appealing kind of science to do, although obviously it's very useful for the public service. But The kind of science I want to do is exploratory, where as long as you can formulate the alternative answers in a clear and cogent, falsifiable way, then either of the answers could be extremely productive.
Right. Right. It's not as though you're trying to make something better or worse. You're trying to figure out reality. You're trying to explore and
Yes.
understand. And, in this respect, one important aspect is the availability of new tools, right? And over the
Yeah.
years, it's just been amazing how powerful new tools keep arriving. So
It's exhilarating.
right?
It's exhilarating. And you can finally ask questions you could not possibly ask before. The way you can actually image live single cells going through developmental decisions in real time. You can play with them. You can perturb them. You can watch what they're doing. You can look at their whole transcriptomes. It revolutionizes what you can say about them, how you can understand how they make decisions. It's extraordinary.
Yeah. And I think, it's extraordinary for you. And I think that's very admirable. I think for many researchers, it's maybe overwhelming, and it's really great. I think we should all have your attitude.
If you live in the questions, the questions are always bigger than the experiments that you currently have the technology to do. So someday, 5, 10 years down the line, there's a technology out there. Suddenly, it can answer your questions. Usually it comes in from a totally different field. It can come in from neuroscience. It can come in from physics. But suddenly it's there, and
Right.
you can use it to get at that question that you've had for maybe 20 years. But you can do it now, and you can pounce. It is a joy. It always has bugs working it out at the first place, but it's incredibly exciting. And, it makes you feel that you never, ever, ever want to stop doing this. It's endlessly marvelous.
I love it. And I
Yeah.
forget who said it, but I think the quote is, theories come and go, but the embryo stays. And
Yes, exactly.
so I think from your perspective, it's these methods, they come and go, ever new, ever more powerful, but the questions remain.
Well, you try to get all the juice out of each method, and then you realize that you can connect it with something new and answer a whole new kind of question. So, yeah, it's a marvelous field to be in if one is lucky to have the support and to have brilliant young people come and work on the problems. I really get excited at the young people who are setting up their own labs and finding marvelous discoveries, and I just hope that they're going to keep on having. These make me feel good about the future, and I really, really hope that they get the support through their careers to be creative and to think widely about the kinds of questions that are interesting that I've been allowed to do in my career.
Yeah, I think that's actually a wonderful way to end by emphasizing how much joy you have in what you're doing, and how much you're hoping to contribute to the next generation of scientists to experience the same kind of joy of living the questions.
Absolutely. Thank you so much. That's a perfect paraphrase.
Yeah. Thank you, Ellen. I think we introduced you as someone with infectious energy for science, and I think that's exactly what we have heard.
Yeah.
Thank you so much. It's been a pleasure.